Just to start off with, um, my name is Smee Travsday. I'm one of the co-captains of Rabbotics, the robotics team on Long Beach Poly High School's campus. Thank you for tuning in to the 2021 Biomedical Professionals Career Panel. I hope you're excited to see the wonderful panelists we have today, spanning biomedical engineering to biotech to academia. But first, I'd like to give some background on our team. So we're our first robotics competition team, for those who don't know. The upcoming 2022 season, which starts in January, will be our 5th year as a group. And our team consists of around 30 students at Poly, as well as some mentors, including our lead mentor, who's from Boeing. During our years as a team, we have sent a robot to the Del Mar Regional, Los Angeles, and Orange County Regionals, and World Championships Houston. The team has also been recognized with the Rookie Inspiration Award, the Judges Award, and as an outstanding STEM program by the Orange County Engineering Council. We have a mission to enable students of all backgrounds to explore STEM and to equip them with the skills necessary to work in teams and to succeed, hopefully, into their careers later. And so if you're interested in joining the team and you're a student at Poly, we are always welcoming new members, and we meet Tuesday, Thursday after school, and you can reach out after the panel is finished as well. Earlier this year in February, we held our first industry panel, which was called STEM in the Environment. That featured various industry professionals in engineering, STEM, and of course, environmental fields. And so if you'd like to view that recording, feel free to search Poly Rabbotics on YouTube. Or visit our website, polyrabbotics.org. So with that being said, I'm going to turn it over to the moderator for today's panel, which is Sydney Chaikitiratana.
All right, so hi everyone. Welcome to our Biomedical Professionals Industry Panel. My name is Sydney Chaikitiratana, and I'm one of the student members here at on the Poly Rabbotics team. And I'll be your panel moderator for today. I'm really looking forward to the panel, and I hope you all find it informative and helpful. So for everyone watching, today we'll have 3 panelists. Our first will be Dr. Daniel Pilloff, the second will be Mr. Jeff Dooley, and the third will be Dr. Siavash Ahrar. So each panelist will have a 15-minute presentation followed by a 5-minute Q&A. So feel free to send in any questions that you may have through the chat, or you can ask them out loud verbally during the 5-minute given question time. And then after all 3 of the panelists have their time, we'll go into a more loose and general 20-minute open Q&A. So you can ask all the panelists any questions that you may have. So thank you again to all our panelists for giving your time, and thank you for everyone for coming. I'm now going to introduce our first panelist, Dr. Pilloff. He has a PhD in biochemistry as well as additional medical school experience. He has taught secondary level sciences such as biomedical research, biology, chemistry, physics, nutrition, environmental science, and marine biology. He also has worked on curriculum development with UCLA's Research Experience for Teachers program, and he is a participant in Cal State Long Beach's monthly physics teacher demo days. So Dr. Pilloff, whenever you're ready, you can go ahead and begin.
Hi, everybody. Nice to see everybody on a Thursday afternoon. It's my screen share was looks like it's disabled. I had put together a very, very small slideshow, but I think you actually just hit all the main highlights. And so, I mean, my actual I thought, because I have to leave a little early, my actual presentation would be on the shorter side, and I would allow more time for questions in the presentation time. So this is my 17th year as a teacher, and my 6th year here at Poly. And thanks, Sydney, you ran through the courses that I taught. Currently, I'm teaching AP Physics and Biomedical Research. And if you can believe it or not, I actually started off as a biology teacher. So if you were to go back to New York and tell them my name, they're like, yeah, I was a biology teacher. So the idea that I'm teaching physics would have been really a total surprise to I think, to my coworkers when I first started teaching. But it's an area that I'm really interested in, and so I get a lot of joy being able to work with students. And I would say that the reason I went into teaching after going into graduate school was that I I took time to sort of reflect on what did I really like, what was I not as interested in, and I really liked learning science. I like being able to continue to do science, and I also like being able to share science. And so for me, teaching was a very natural career path. I had the advantage, actually, a roommate a couple years ahead of me who had gone into teaching, so I could see a little bit behind the curtain of what it was like. And so I've been very, very pleased with the career. You did mention when I started that medical school background. So when I finished college, I was I double majored in biology and chemistry, and I was applying to what are called joint MD PhD programs. And so I got into several, and I went to one in New York City. But I discovered in my first year of medical school that I just wasn't enjoying the study of medicine as much as I was the more basic sciences. And so I just kind of had this moment of realization that I could continue in this program, but it definitely wasn't something I was enjoying, and I didn't want to be miserable in a career doing something that wasn't fun for me. And there was this very, very closely related career in research that was really, really super interesting. So it's a little bit of an uncommon switch, but I sort of dropped the MD part and just went solely PhD. And that for me was very, very intellectually interesting. And since then, I feel like going into teaching has just been a very natural career path. And increasingly I think a fairly common career path for people who go to graduate school. That there's now what's called, it's a term you may or may not be familiar with, but it's been kind of codified as what's called alternative science careers. And it's basically from the just the raw numbers that most science departments take on each year as graduate students, more graduate students, than they have basically faculty appointments that they would need to fill that year. And so we are, I mean, as a science education industry, we're training more people in high level science than there are positions available in academia. And so some people are really, really set on working in academia. And so it's, to take that path is a bit of a gamble. There's really no guarantees. When a person starts applying for academic positions, that they'll land one. And then the other aspect of going into academia is a person has to have a bit of a lifestyle flexibility of being willing to move to other parts of the country. One of my really good friends in college actually took a job in Singapore at the Singapore National University for his first position in academia. So this sort of matching of personal interests and career opportunities and how you get there does have to take into account your own sort of style, your lifestyle needs. And for me, I really wanted to be in New York. That's where I met my wife. She's actually a really important part of how I got to Long Beach. She was living in New York at the time, and so we met there. And eventually we decided it made sense for us to move closer to her family in California. So that's how I got here. But certainly, we didn't want to entertain the idea, that would be moving to somewhere, who knows where I got an appointment. So I've been very happy with teaching, and so I think I've been talking maybe 5-6 minutes. If there are more specific questions, I'm happy to take them, or I can talk more about kind of the curriculum development stuff I've been doing with Cal State Long Beach and UCLA.
Yeah, if you want to elaborate on that, I think it would definitely be a good thing for us all to hear and learn about. So yeah, go ahead.
Well, I mean, one of the really cool things about teaching, and I saw this both in New York, and I saw it certainly true here also, is if you go into teaching, there's usually a lot of partnership and collaboration opportunities with other educational institutions. And a lot of those are universities. There are other more sort of grassroots organizations as well. There's this sort of called citizen science. I don't think that's necessarily even an organization, but it's more of a theme that, you know, people just sort of step outside their doorway at home, and they find a flower or an insect or just something that's intriguing to them, and they share that with other professional researchers. And so there are these there are these programs you can upload a photograph of a flower that you found interesting, and you know, a box will it's sort of in the meta universe of professional researchers. Somebody will identify what that was for you, and so it's just, there's a lot of opportunities for people to engage in science, and it doesn't have to be only with the university level. But for whatever reason, I was sort of connected. That's probably the best way to say it. I was connected with some opportunities. One at UCLA. So they have a program called Research Experience for Teachers, and so I was working with 3 other teachers on developing curricula to try to develop new labs and new activities for students to learn about electricity and magnetism. So in the past, I've taught AP Physics C, electricity and magnetism. Hopefully in the future, that may also occur again. And so I was developing some labs with them. And more locally at Cal State Long Beach, they have a program where they kind of recruit local physics teachers to go to their workshops once a month. And we get to interact with the current physics education students. And so sometimes it's teachers sharing their demonstrations, which I've done. And sometimes it's more of there's like a hands-on project that all the teachers will work on to sort of learn a new trick. And then you learn tricks just from seeing other teachers' demonstrations. So actually it's one of my favorite, it's one of the highlights for me each month, being able to take part in that. And you know, even virtual, we've been continuing that. So we're doing our demonstrations through Zoom videos. So it's just it's sort of the nourishing water that for me is very important.
All right, Dr. Pilloff, we do have one question. So you mentioned earlier you completed some medical coursework. So what would you say to those who are interested in pursuing maybe a medical degree, and what are some alternatives? Maybe acknowledging, obviously, like for yourself, it may not be for everyone.
Yeah. So I started off in the MD PhD program. Just for people who don't know a whole lot about medical school, if you might have some preconceptions / misconceptions, at least at the time that I was applying, and I'm pretty sure it's still pretty strongly true, you don't have to be a science major to apply to medical school. I had heard that nationally, maybe only roughly half of the students who go to medical school are science majors, and now that's probably encompassing all the different medical schools. Many of the ones that are more local to here that you've probably heard of are more major research institutions. They probably skew to having somewhat a higher percentage of students who did major in a science. But at my at the school where I went, the Albert Einstein College of Medicine, I had, I think, both of my roommates who were medical students. Neither one of them was a science major. One was a history major, and he became a psychiatrist, and the other may have been a major in government or history. He might have been a biology major. It's also just to kind of bear in mind that, you know, for students who are, sort of ambitious, which is probably a personality trait of people who go on to medical school. Many of those students probably will have done 2 majors, and even if you're not going to go. So I was a major in biology and chemistry, so it's fairly common now for universities to have some pretty well developed pathways. So for some majors that kind of pair well with each other, biology and chemistry is a common one. There was a classmate of mine at Einstein in the MD PhD program, and he had kind of an unusual pairing. He went to Berkeley, and he was a double major in biology and also electrical engineering. he was actually a really, really stellar MD PhD student. I think he had something like 20 publications by the time he had finished just graduate school. And, so, but there's many different on-ramps to going to medical school. But what I would say is that before you go, you should have a pretty clear idea of what the what the nature of the work is like. And so I actually had a pretty good idea of. I mean, I kind of knew what I kind of knew what the courses would be that I would be taking. What I hadn't really experienced was the actual experience of taking the courses. And I just found that once I started taking, there's a lot of, in medicine, there's a lot of stuff that you just have to memorize. And I mean, perhaps if I had gone to medical school right out of high school, I sometimes wonder if I might have found it actually more enjoyable. But just because of the sort of intensive science classes and research that I was doing undergraduate, I just found that the way I was thinking and what I found interesting sort of shifted, and I hadn't really realized it. And so in the medical school courses, I wasn't really so passionate about the tremendous amount of memorization, and I just sort of realized that there's no way I'm going to do this as a career. For people who don't know, most of the combined MD PhD programs are paid for through somebody other than the student doing it. So you're basically getting an 8 years. Some students take 9 years, depending on how many years they spend on their PhD. You're basically at most schools, certainly around here, the program is paid for. That being said, there's also an argument to be made that you shouldn't do an MD PhD program simply for the free medical tuition, because you are adding on 3, 4, or 5 years of additional time that you're working before you're really sort of really getting a full salary. I mean, so when you're a student in an MD PhD program, you're basically getting a stipend that covers your rent and your food, but you're not able to really save money in any sort of meaningful way, in the same way that when you would have a job. I mean, at the time your expenses are fairly minimal because you're working a lot in the lab, but it is kind of important to mention that that most of these programs do offer sort of the financial trade-off that they'll pay for your basic living expenses. You're putting in 4 or 5 years extra, perhaps, if you're doing your PhD. I mean, some students finish the PhD in 3, but the part about the PhD work in an MD PhD program is it's highly dependent upon who your advisor is, what their own particular philosophy is for publishing. So my friend who had 20 papers, I mean, he had an advisor whose philosophy was publish fast and then publish fast again. And then there were other advisors who wanted to publish a little more slower, put together sort of more comprehensive papers, and the opportunities for that also probably depend upon the field. So most students, in my sense, I get the sense that there's a few more students in 4 than 3. Finishing in less than 3 is highly unusual. There's a good number that probably do 4.5 or even 5 years. And then the medical school, I think the medical schools usually have a little bit of flexibility. Like the 4th year of medical school, there's a lot of flexibility. They're doing electives, and so sometimes they cut 1 or 2 electives out of your program just so you can kind of graduate by a June deadline. And then after you finish the program, the pathway usually would be you would apply to residency programs. So I did not have any experience applying to residency programs, but my understanding is that MD PhDs are usually fairly competitive for that. And then after residency, I mean, some MD PhDs will go into clinical practice, but a good number of them then go into academic positions, and there may be a few other stages along the way. So, so my friend who is and cut me off if I'm running into anybody else's time, but so my friend who gets something like almost 20 publications. I think he graduated in 2004, perhaps. maybe 2003, 2004. He would have finished his residency probably around 2007. And so, at this point, he has a lot of papers, but I think he then had to then work basically as some sort of fellowship post residency. And so there was probably like another 5 or 6 years from the time he finished his residency till when he got his university appointment. But he did get it, and so he's at Washington University, which is a very, very well-known research institution for medicine.
All right. Well, thank you, Dr. Pilloff. We have a couple questions from the chat. So if you wouldn't mind answering those.
Yeah. So I was looking at the chat, and so those questions must have been sent to you privately.
Yeah, they were direct messages.
Yeah.
So I can read them out to you. So would you mind elaborating on some of the research you've done either during your medical coursework or at some other point, and what was the research process like?
Yeah. So usually, if you're getting into research, I got into it in high school, and the sort of your on-ramp into research in high school is a little different than in college, and definitely different than as a graduate student. So I'm from the East Coast, and it just happened that where I grew up in the Washington, D.C. area, there was a well-developed program for high school students that probably took, I'm going to say, probably like 500 students each year into laboratories that were run by the Defense Department. And so I did research at the Naval Research Laboratory. I was not the only student there. I think there must have been like 100 other high school students in the Naval Research Laboratory. And then there were many other labs where students went to. And so when you go in high school and you don't really know anything other than what you've done in your high school classroom, there's a pretty steep learning curve. You usually get paired. The person of record who's your mentor is probably not going to be the person who interacts with you in the day-to-day a whole lot. They'll meet and greet you and kind of introduce you to the big picture, and then there are other people in the lab who's postdoctoral fellows or research faculty, which is kind of a stage beyond postdoctoral fellow, who they work with you more closely, and you'll usually learn 1 or 2 or 3 different research techniques, and you sort of in high school you're sort of just working on perfecting that. So the scope of a research project for high school students is usually fairly narrow. It's more about just learning how to do things reproducibly and understanding the background process, and spending a lot of time learning how to read papers. And so by the time you finish that experience, you kind of know you've learned something, and you sort of see how this fits into the big picture, and you can talk about it to other people. In college, because you've already had now some further science courses, doing research becomes more like sort of like a year-long commitment, and that's also where there's generally a lot more choices of who you can work with. The truth is, there's very few labs that actually will take on high school students. It's kind of a sort of a unique opportunity, but in college it's actually fairly common, and so there's a much wider opportunity to sort of search out different areas. And so that's the stage I would say that if you're going to do research in college, do your homework, and what that means is probably reading papers that the researchers have written. You don't have to go through it like with a fine-tooth comb. Like you're not going to become a master of their research, and that's what they are. But see what different people are doing and think about what you're interested in, and probably also, if you have a good college advisor, ask them for advice. I would, so take advantage of all the resources, even if some of them you have to sort of lift up some stones to look for those resources, because you're basically making sort of a longer term commitment. And then of course, if you have a good research experience, I don't want to make this sound like the sort of transactional, but if you have a good research experience, certainly those mentors could be very supportive in terms of applying for graduate school. And I would say that is certainly an expectation when people do apply to graduate school. From what I've read, those letters from the mentors carry a lot of weight. So if you're going to apply to graduate school, probably, I mean, supposedly the standard is 3 letters, but I suspect people are doing 4. I mean, sometimes you probably want like 2 professors in academic classes, 1 solid mentor from a mentor, and then maybe some people are doing a 4th letter, squeezing it in somehow. And so the more invested you get into it, kind of the harder to extricate yourself if you want to change directions. People change directions all the time, but it's easier to change directions as a high school student than as a college student, and then even less easy as a graduate student, where oftentimes you've been accepted to a certain department, and if you want to change directions, it can be done, but it requires a lot more coordination with perhaps a director of the graduate school, or a committee of different professors if you've already assembled. So you want to just try to explore your options, and kind of know what you're getting yourself into.
All right. Well, thank you, Dr. Pilloff, so much for your time and your expertise
course.
and everything. I'm not sure if you have to leave early, but you can go
I
ahead.
do. My wife is working this evening, so yeah. So I mean, I can take. I could stay for a few more minutes if there are more questions, but I'm kind of sensing that that phase is past. But I won't. But I actually have to be. I've got to be home in an hour.
All right. Well, if you need to go, we're good. We can keep going from here, but thank you
Of
for your
course.
time, and we really appreciate it.
Sure. And feel free to share out my email. I'm in room 260.
Yeah, of course. Thank you.
Of course. Okay. Thank you for putting this together.
Yeah. So we're going to move to our next panelist. This is Mr. Jeff Dooley. He studied materials science and engineering at Cal Poly San Luis Obispo and works on diagnostic equipment design. So Mr. Dooley, whenever you're ready, you can go ahead and begin, and I'll.
Sure. Thank you for having me. I'm pretty enthusiastic about this. I see you've got over 20 participants, so I hope this is helpful to somebody. A little bit about myself. So I've been a career engineer for getting close to 20 years now. I've grown up in Southern California, and I've lived in Southern California my whole life. Went to high school in San Diego County, and as you noted, I went to college in San Luis Obispo, and then I've worked for a number of different employers, but everywhere I've worked has been in the local area. So let me just go back to the prompt. So yeah, I mean, a little bit more about me. I've got 2 kids. A lot of them are close in age to you guys. I have a son who's a middle schooler, and I have a daughter who's a 9th grade freshman. So I can kind of relate to you guys a little bit because anyway, I get a lot of questions from my own kids. Let's see. So currently I work for a company called Microvention. We're actually a subsidiary of a bigger company, a big kind of conglomerate from Japan called Terumo. You may or may not have heard of them, but anyway, that particular healthcare company services all different facets of the healthcare industry. So whether it be hospital products, whether it be vascular devices like the ones I work on, or diagnostic equipment or other things, that bigger company, parent company, owns a lot of smaller divisions like our subsidiary that kind of services almost the whole healthcare market. Okay, so I'll give you a little bit of background, I think, and then kind of get into the what I do on a day-by-day basis. I think hopefully I kind of started out a little bit like you guys. I was interested in biology from the very get-go. not until my senior year, actually, did I really get, make a change in my my overall plan. I thought I would pursue a biology track and maybe go into medicine. Potentially, I think when I was a junior, I was thinking I would maybe try to do immunology and try to do laboratory work. And then I decided my senior year, no, I think I'd like to be an engineer, because I'm a lot more mechanical by nature. I really like aspects of mechanical engineering. My degree is in material science. So if you don't know what that is, it's kind of like solid state chemistry. So I didn't like wet chemistry so much, of mixing of solutions and reactions like that. Rather, I liked solid material properties and things like that. So like in aerospace, they'll hire folks that do material science to work on super alloys for space missiles and jets and things like that. Really cool stuff. In the medical field, we also use pretty unique materials because they're radiographic. You can see them under X-ray, or they're really springy, or they just have kind of unique properties. So we're not looking for just some run-of-the-mill plain carbon steel that just you can run gobs and gobs of it for free. We need materials that will function in an application that is really precise. So that's kind of like my educational background. Also, I guess I'll throw out there that I would say I was probably kind of a okay student. Certainly wasn't the high flyers. I wasn't your 4.0 plus group. I didn't go immediately after school to pursue an advanced degree. I did get an advanced degree, but several years after I had already left college. So yeah, so I think I went to an okay school, and I think I had an okay educational background, but that's about as far as I go. But yeah, I started off just doing design engineering. And really, although my education was in material science, actually right out of the gate, I didn't use it at all. I got hired by a startup company. They needed somebody who could do anything. So, so I did a lot of testing and a lot of things that you wouldn't necessarily say you went to school to do. But I got a whole lot of experience, and a lot of that experience included design work. So, so I saw, I think further down the prompt, things that I did right out of school, or I should say right out of college, would be things like CAD, or some, I wouldn't say advanced statistical analysis, but maybe some kind of basic modeling. So when we were trying to do design, we want to know before we build the part whether the would break. So we would try to do some strength analysis, or we would do some, I don't know, just some kind of cycles, cycle predictions, whether something would, if it broke at a 10-pound load and it broke after 10 cycles, if you drop the load to say 2 pounds, would it go 100 cycles or something, just some basic predictive techniques like that. And just using spreadsheets, not using anything advanced. Yeah, so I did that for a number of years, and then life happens, you get married. You maybe don't want to work 80 hours a week at a startup. You maybe want to take it relaxed and work at a bigger company. So I've worked, in total, I've worked for different companies, like starting at, Blue Sky startup. I've worked at major conglomerates like my current employer, and doing all kinds of different things. As I say, I started off in design. I really got a really good background in design. I've done manufacturing, I've done quality, I've done, kind of variations on those things. Currently, I'm in a leadership role. I've been doing leadership for the last 3-4 years. So I'm a project manager. I've got a team of about 10 engineers and techs, and those engineers and techs are working on 4 different products we're working on. They're kind of in different phases of the life cycle. So we've got 2 products that are commercial now, but need improvement and things like that. So we have engineering projects to either improve yields or, make less costly. We also have new projects that are in development. So we're doing early R&D phase stuff too. That's kind of what I know most of. It's kind of what I've done throughout my career is kind of new development. So I'll just kind of touch on what I think are the things that you guys might like to know about. Yeah, so like, at least from what I've seen, the major categories that drive design are clinical need, or some new clinical application or some therapeutic application that, technology is now enabling. So there's some research into clinical application. There is technology, which to me is normally a processing technology. So, I laugh at my supply chain friends because sometimes they say, oh, can a laser do that? And lasers don't do everything, but lasers are some things that they can do. So a lot of processing technologies, right? So to either to image or to cut or to, build up or, a lot of folks now are moving to additive technologies that you've probably heard about in your robotic stuff, right? So 3D printing, not only for plastics, but also for metals. I haven't yet used a 3D printed metal yet. I use a lot of 3D printed polymers, but I'm still looking for my first metal. And then of course we have something that's more aligned with my educational background, which is like the materials properties. So in healthcare, or at least in the products that I work on right now, we're really pushing the edge of what you can achieve in a material. You can't do bolts and screws and big mechanical designs. What you try to do is optimize your properties of your product by altering the material. So not sure if you guys know what a catheter is. It's a long, slender product that is advanced into a small part of your body. Either what the body that we treat at Microvention is part of the brain. So what we do is we implant something that helps block and prevent a rupture in a blood vessel. We call it an aneurysm. So if there's a little pocket in your blood vessel in your brain, and it's basically a weak part in the blood vessel, we basically clog it up with an implant. Or the other thing we might do is there might be a blockage, like a clot. So I don't know if you've had any family members that have had a clot or a stroke. That means that there's already a blockage there, and we want to try to get it out as rapidly as we can. So for stroke therapy, we actually put, for sake of simplicity, a hose up to the clot and suck it out. And so because the brain is so tight, we actually alter the properties in the material and not try to bolt on new features of the product to make it complicated, because it won't work. It will never get there, never be usable. So kind of coming back, so to me, the high level things that really drive industry forward and technology forward are clinical needs and clinical applications that are new and different and things that have never been done. Like processing technologies that have never been done, either imaging or, any number of things, nuclear, who knows? And then materials. So those are the kinds of things that I've kind of worked on. I've never done a nuclear device, but anyhow, let's see here. Ending summary. Yeah, one of the things that I think is a major takeaway for you guys as kids, maybe the single biggest takeaway, is do something that you really like to do, and don't necessarily try to know what that is today because you're 16 or 17 or something. You're gonna figure it out. But it will take time, and just hold on to that, because what I have found is that bottom line, here's the bottom line. If you don't like doing it, you don't like doing it. And the converse is, if you do like doing it, you love doing it. And I feel totally blessed because what I do is what I love to do. And I've really had a fulfilling career because of it. So I guess I'll maybe just kind of drop off there and then let anybody ask questions if you have them.
All right, thank you. Hamid, do you have a question? Or if not, I have some in the chat that I can ask.
Let's start with the chat. Yeah.
Okay, so the first one is, what do you like most about your job and what are the downsides?
Well, I mean, so you guys haven't been in the working world, right? I mean, maybe you have had some jobs, but every job is a job. I don't know how to really put it more articulate than that. Sometimes people are people. So there's usually if there's something that you dislike about your job, it's because there's something people related. One of the things, so I'm, now I'm kind of in a leadership role and I'm very people oriented, and which is fine. I mean, I like people, but yeah, that's usually what you find is if there's a downside, it's because somebody has a personality thing, or, people are people. Sometimes you like them, sometimes you don't.
All right, thank you. And then Hamid, do you want to ask your?
Yeah, so you kind of touched on this, the people side of things. As a robotics team, especially in the coming weeks, we have to build a robot in 6 weeks. So I just want you, maybe if you can elaborate, like what's the project management process like in engineering? How is teamwork part of your job? What does that entail?
Yeah. Yeah, I mean, how to synthesize this? Like for robotics or another technology project, usually I think what often happens in a team is you get a lot of smart people together, and everybody thinks that they've got the solution in their pocket. They're like, I have the answer, here it is, and go do it. And you have some folks that are kind of quiet in the background and don't want to say anything, but some people are really loud and boisterous and excited and just can't share their ideas. And usually it's trying to find like an even flow, like trying not to get to the end all in one big giant step, but involving the team at large and try to progress in kind of a logical fashion, like you're not trying to do it all too fast. Everybody wants to do the big giant achievement, ta-da, it's done. But it never works that easily. There are kind of some steps involved, and seeing the staircase as opposed to just one big end goal is kind of how I'd answer that.
All right, thank you. The next question we have for you, probably the last one for the specific questions, are what is the future of the biomedical industry, and what would you like to see young people like us pursuing?
Okay, let me get my crystal ball out. It's over here somewhere. I mean, to me, things that are like on the like the very the horizon, I should say. Artificial intelligence. There's going to be obviously like other industries, we're employing a lot more software. So our CEO was talking about how he would like to have our next generation products to integrate with your cell phone and other things like that. And like for me, that's like oh, who cares about all that software stuff? But definitely, I think medical therapies continue. I think there's a lot of future to it. There's there's always a kind of like an inherent battle between the pharma people that are looking to make a drug or a biologic or something that you can kind of just take as a pill, versus somebody more like me who works on devices. But you know, we're all working together, of course, to improve life. And yeah, I mean, it's hard to say where exactly it will head, but you know, just kind of knowing what new things are coming, come not just to one industry, they're going to come to all industries.
Okay, great. Thank you for your time and for
My
all
pleasure.
your information. Now we'll be moving on to our last panelist of the day, Dr. Arar. Sorry, hold on. He has a PhD in biomedical engineering from UCI and was a postdoctoral researcher in the UCI Department of Physics. He was also a Science and Engineering Education Fellow, and he is currently working on a curriculum for the discipline of bioengineering. So, Dr. Arar, whenever you're ready, you can go ahead and begin.
Great, thank you so much. Thank you for the kind introduction. Is it okay if I share my screen to show you a couple of hastily put together slides? I apologize if these are not very well put together. So unlike the previous speakers who have a tremendous amount of insight talking with younger folks, my weakness is that you're the first group of high school students that I've actually spoken with professionally or personally in a very, very long time. So I've been very nervous and excited about this talk, because I always tell my students that having empathy for our audience is the most important thing we can have. And I don't know high school students, so feel free to stop me at any time if anything's unclear. I've got really set to speaking academically, which is never a good thing. So my goal for today is to talk about just very little actually about myself, but mostly about tons of opportunities and exciting things that are available to you in the near future if you choose to pursue a technical education at the intersection of engineering and biology. So my claim is that you are living in the golden age of biology, essentially, and biological research. The same way that perhaps maybe 20 years ago, folks would have been able to say you're at the forefront of amazing things happening in computer science, which continues to happen. We have been living in an era that biological discovery, the rate of actually automation and engineering, because of things that folks from an engineering background have brought to the discipline of biological research, has really transformed that field. So here on the right side, there are two photos of the same system that we recently have built in my lab in collaboration with a friend at UC Irvine, where this gizmo is a very simple do-it-yourself microscope where you have a bunch of 3D printed parts. There's a laser pointer actually to follow up with Mr. Dooley's description that lasers can't do everything. We use lasers to excite molecules that shine light back so we can see them essentially. And here you have the objective, and there's a little glass slide that has microfabricated channels inside them, channels that are about hundreds of microns. And if you remember, micron is 10 to the minus 6 meters. It's about, 100 microns is about an average size of a human hair. And the reason we built these tools, we built these systems, is because they enable us to grow, cultivate, keep track of biological systems in a very controlled environment. And I'll show you examples of this in next future slides. Just very briefly about my background, unlike most of you who are, as high school students, are super excited about, I think, STEM field as a whole. Even though, so I did not go to high school in the United States. I grew up overseas. And over there, we had to pick a major actually for high school. So I was a science and physics major. I was really good at math and physics. But I actually did not want to be an engineer or a physicist, or I had very little interest in STEM as when I was your age. And it was actually typical in my family in a surprising way. I wanted to be a writer. and there were 2 writers in the family, so the precedence was like higher for me to be on that track. And as time went on, when I joined college, for example, I started to get more invested and attracted to engineering. And from a naive perspective, the way that I imagined being an engineer was about building stuff. That's really what I imagined. You know, I'm not really a professional engineer. I've never worked in the industry, but from the academics point of view, the way I see what we do is that we design, build, and test, and iteratively try to improve a system so that we can accomplish the goals, the specifications that we set out for that particular system. Now, the flavor of engineering that we work with are the things that have to work with biological systems. And biology is super beautiful. We can begin to think about biology as a technology, which is both a very promising idea and also it's a bit of a scary idea. This is something that you have to live with for the rest of your lives. For example, the field of tissue engineering promises that we might be able to 3D print organs and be able to replace damaged organs. That potentially will be feasible, maybe not in my lifetime, but in your lifetime, like give or take 20 years difference. That's a big difference in scientific advancement. That might be something that is feasible. So since I'm an academic, I'm going to give you a homework that you're going to do right now as we're going through it. So I want you to imagine a biomedical or bioengineer. For our discussion, there's no difference. And just giving credit, this activity is from Professor Sherry Shepard's group. So I just want you to begin to put down, so that I can also build empathy towards high school students, just put down a couple of adjectives that you might think that describes a biomedical or bioengineer, things that just pop up to your head. And feel free throughout the rest of the time that I am chatting with you, please try to draw a cartoon diagram, a doodle of a depiction of an engineer, someone that when you think as an engineer, that's the image that comes to your mind. So that's your ongoing homework for this little activity. And I'll show you an example that from actually a friend's drawing, because my drawings are horrible. And I'll tell you why we actually do this activity in Introduction to Bioengineering at Stanford and Introduction to Biomedical Engineering at CSULB. So it's getting the first day treatment that folks in our classes are getting. So I'm going to show you at first 2 examples of folks that I look up to as a biomedical engineer. So when I was an undergraduate student, these were the professors that I wanted to be like, and I've had the pleasure of meeting them both. So this is Polly. Polly is a biomedical engineer, and in her lab, they built these microfluidic devices. So the idea with microfluidic devices can be simplified as chips that have microplumbings on them. That's really what they are. They're silicone rubber chips, like they actually feel like an eraser, that have channels that are hundreds of micrometers in width and height stamped into them. And you can run various chemistries or diagnostics throughout these chips. So if you want to run, for example, it might have done this in your chemistry or science courses, run a reaction, you have a big beaker, you add various chemicals, and you're trying to keep track of seeing like how the color of that reaction changes, right? So because of the size and the volume of that reaction, the reaction is going to run slow, and you get one outcome based on that large, volume that you're running. So if you make everything smaller, the reaction times will go faster, and you can kind of look at many reactions simultaneously. So here, if you're taking a look at photo on the left, where they say these valves and sandwich button, the idea there is that folks like Polly have built tiny valves, tiny chambers that open and close when you apply pressures, like 10 psi of pressure, and they can move liquids that has biological samples inside there. And now they can run 1,500 reactions simultaneously to look and see if these proteins react with one another or not. So if you want to do biological discovery, this is one way that you set out to automate or apply engineering principle to the discovery of biological systems. Does this make sense? Any question about here? So the color that shows that, there's an arrow that says time, the places the chambers are getting brighter are the chambers that the reaction is happening faster. So if you're looking for a protein that is reactive to its substrate, the middle chamber is your target. That's the winner, so to speak, for the system that you're in, biological system that you're looking into. Another person who I looked up to when I was genuinely like a little older than you is Professor Manu Prakash. And Manu is a very, very interesting scientist. He advocates for frugal science and that idea of citizen science, essentially. So his lab has been very good at building systems that enable everyone to engage with research. So this thing that is on the photo that is next to Manu's photo is called a Foldscope. It's a paper origami microscope that you can get actually relatively affordably from their website. I think it's $5 to $6. The shipping is actually more expensive. We get this for all of our students in biomedical engineering, introduction to biomedical engineering. And under the photo, you can see an example work that is produced by someone who's maybe 1 or 2 years older than you, that they were just walking around and actually they were interested in material properties of this biological specimen that they found. They were wondering why is it squishy, if you will. So they broke it up and look at it under the microscope. So the Foldscope is a very interesting tool. Your mileage may vary. Some people really like building it. Some people really like taking photos with it. So it's a tool that you can use to engage with biological research, essentially. And recently, actually, Manu's group has put out this very neat, completely open source system that is called and I'm going to mispronounce this word, Scope. And it moves pond water, essentially, bunch of tubes and bunch of pumps, so that you can use a very, very simple Raspberry Pi controlled microscope to look at pond water to see if you see planktons. They are sending these to go to Antarctica so that they can look at various organisms that are living around, in extreme environments. And the reason this is nice to have, because if you have one of these super fancy, very big microscopes, they're really horrible for building. They're built to be parked inside someone's lab and never moved again. But this thing, it costs like, $2,000 for a research project that's quite affordable, and you can build tens of them. And if one breaks, it's not a problem. You try the other one, or you can rapidly fix it. So these are two examples of engineering systems applied to biological research from academic setting. Here's another tool that I think you would appreciate a lot. This is an example of a robotic tool built for biological discovery in industry. And I have no financial interest in any of this, just declaring that so that I don't get in trouble down the line. So this machine is called an Opentron, and it's essentially a 3D printer that moves a pipette in X, Y, and Z. So you folks are roboticists, you have a much better understanding of how any of that works than me. But essentially, it turns out that you can be an undergraduate scholar in someone's lab doing biological study, and they give you a pipette, and your job is to move this liquid all the time from a transparent tube to another transparent tube. And it's cool, actually. It can be really exciting, but it's a tedious process. As engineers, we should look at that and say, I don't like that. Can I make that go faster? Can I make it be more reliable? And here's an example of a system that exists today that make this faster, more reliable. So you can use a very quick Python script to make this thing automated, and this will run all of your experiments for you, essentially. So I showed you an example of works that other folks have done. So I'm going to very quickly show you an example of a couple of things that I have done. So here is another kind of like similar to work that I showed you with Poly's system, that we are stamping these chambers and channels in silicone rubbers to grow and keep brain slices, mouse brain slices alive outside of the, I guess, the rodent's body for biological research. So it turns out if you make chambers and channels that are tinier, that are the size of a human hair, forces that we are normally used to, inertial forces, right, are no longer dominant, right? So mixing of liquid, if I get two beakers and pour one colored liquid on top of one another, I see the two liquid rapidly mixing with one another. But if I make the beaker tiny, viscosity, the friction between the two fluids, become the dominating force. So they become essentially like honey flowing next to honey. The advantage of that is that you get boundaries between those two fluids moving next to one another that are sharp. So the fluids will not rapidly mix with one another. So this is shown over here, perhaps with a cartoon diagram, and also the actual image of the device where we are literally using food dye to visualize the boundary between these two fluids flowing next to one another. So we particularly use this system to deliver various drugs to known regions of a brain inside a brain slice of a mouse to study and see how that drug interacts with various regions by being able to block the activity of the drug by removing the drug rapidly and creating sharp boundaries between those two fluids flowing next to one another. Here's another example of similar system. So this was intended as a diagnostic device. So here, there's a bunch of tiny valves. They're about 1 millimeter, actually, each of these circles, that are moving two fluids through a sequence of steps to dilute the concentration of a chemical, in this particular case, a blue dye. So hopefully you can see that the darkness of the chemical is getting reduced. So now I have a concentration curve of various amount of molecules that are inside that chamber. Actually, believe it or not, we were trying to build this to be able to run this assay called LAMP that is currently used a lot for COVID diagnostics. It turns out that the fluidic chip worked very well, but the assay in the fluidic chip did not work. So we had to just publish this part of the system. Other examples of building these types of automated fluidic systems include building kind of like chips. I'm just going to apologize. This is very small. I'm going to make this a little bigger so you can see. These are pneumatic systems. These are essentially very similar to computer chips. If you imagine a transistor that you might have used in your robotics components, switches that open and close with application of voltage. These are similar to them, but they open and close with the application of air pressure. And we've built kind of like oscillators and counters that run on air as opposed to running on electricity. If you're interested, I'm more than happy to tell you why we have done this. We also have used similar technologies to study how certain bacteria interact with patterns on a surface that has pillars that are in nanometer scale. So nanometers 10 to the -9 as opposed to 10 to the -6 meters. The idea here is that can we make surfaces that certain pathogens cannot get attached to? So actually for the system that Mr. Dooley was talking about, catheters, the idea here was, can we pattern nanostructures on potentially catheters so bacteria cannot get stuck to them, essentially. And actually, this is a work that I've done with my partner. So she's the lead author, giving credit where credit's due. She's the lead author on this study. Perhaps what I'm best known for amongst my friends is actually this story, where when I was a second-year graduate student, I've always been a comic book fan. I'm sadly a DC fan. I think that Marvel these days with your generation is far more popular, but I've always been a DC fan. So there was this running joke when we were talking that Batman always has these very awesome gizmos that he pulls out of his utility belt, and there's no way that those stuff will fit inside his utility belt. So as a comic book fan, I just one time genuinely got fed up and wrote a letter to the editors of DC Comics saying that, hey, maybe you want to look into this thing called microfluidics, which is the stuff we built for diagnostic, for chemistry tests, so that if Bruce is detecting Joker toxin and the thing that he's pulling out of his belt looks like a giant system, that just doesn't make sense. The technology for what you're trying to do exists. This is what it's called, and this is how you would use it. And they were very generous to publish my letter, but they never took us on like being able to explain to them how they can implement that as inside a story. So I've been among my friends been known as the comic book guy. So that's perhaps my most biggest accomplishment throughout the grad school. So I'm gonna close with one thing that I tell all of the undergraduate scholars that join my lab. So one of my roles as a researcher is to actually train and mentor folks that are perhaps a couple of years older than you. And this is the first paper we always read whenever someone joins the lab. And it's called The Importance of Stupidity in Scientific Research. Now, I'm not a scientist, right? I'm an engineer researcher. But the claim of this paper applies to my discipline as well. The claim here is that when you try to learn something new, it's a bit scary and you will feel stupid. It's just bound to happen. You can react to this two different ways. You can react to this by retreating, or you can be productively stupid. You can say, well, yes, this makes me feel bad. I really wish that, that I wouldn't feel bad that as I'm trying to learn this new thing. But if I acknowledge that, okay, that's right, I can try more and I can have this growth mindset that say, well, if I ask for help, like if I ask for feedback, if I try again, I can improve. If I really care about learning this, I can improve and get better. I can proactively, iteratively, we're engineers, we're in design, build, and test, right? In learning, we can also try to iteratively get better. So we always read this as a group and we really believe in it. So we have this printed and placed on our wall. And sometimes we jokingly say, do you feel stupid today? And if not feeling stupid today, that's a bad sign. It means that maybe you've got a little comfortable with not trying to learn more or try to move your project further. Now, I'm not saying just do stuff randomly. I'm saying let's be proactively stupid. And this is not my idea. This is, I think, Martin Short's idea. And it's a very, very good mindset to have when you're trying to learn or build something that no one else has done before, or even if you're doing something for the first time. So yesterday I was working with this system and I really messed it up. I felt really stupid. So I asked my collaborator, who's the microscopist in the team, I said, hey, Nick, I messed this up. Can you show me again how to focus this thing? And he was very generous. He kind of like ignored the fact that I almost ruined our experiment. He came back and fixed the system and showed me again. So I'm looking forward to messing it up in the coming weeks. So I started by asking you to do a depiction of what do you think about when you think about an engineer? And this is a drawing from my very good friend, Veronica. Veronica is an environmental engineer, where she uses genetic tools to see sequencing, particularly to see what are the microbial systems, some of them harmless, that might be inside a water plant. And these are two images that Veronica has drawn over a period of, I think, 3 months. And the major difference actually between these two images, I think, is the complexity in a good way, or kind of like representation of a lot of ideas that are on the picture on the right. The picture on the right is the second picture, right? After thinking more and trying to proactively be an engineer, right? So here, on a first approximation, an engineer can be a problem solver, creative, can be logical, and those are all true, right? But also an engineer can have a growth mindset, right? An engineer can be, as Mr. Dooley was saying, working with nice people is nice, right? Engineer can be nice. Engineer can have empathy towards working with systems, right? As someone who works with biological systems, I am acutely aware that we touch stuff of life. So it's very important for us to have empathy for the systems that we are working on. So I wish for you, I'm not trying to advocate that try to be a better you're good at drawing. My wish for you is to try to think holistically about whatever career that you pick or whatever trajectory that you choose, right? It's good to try to sometimes take a step back and be like, well, what is it that we are doing as engineers, or in my case, bioengineers, like, and try to have a good sense of where we want to go with our discipline, essentially, and how we interact with society. So thank you so much for your time. Hopefully this was useful to you. I honestly cannot tell because, as I said, you're the first group of high school students that I've ever spoken to. Even as a graduate student, I've spoken with actually retired folks. That was like probably the most engaged audience that I had. Like as grad student, you TA, and you sometimes wonder, like, okay, why aren't the students paying attention to me when I'm telling you that this is literally on your test? But retired folks were amazing. So they asked so many good questions. So I don't know if this was useful to you or not, but I'm more than happy to answer any questions that you may have.
That was an awesome presentation. We do have a couple of questions here. This is research focus. I think one person is saying, what's the purpose behind making the pneumatic transistors?
Right. So it turns out, and I'm highly biased in this. So let me just proclaim my bias because that's my PhD thesis project. I feel less emotionally attached to other projects that we've done. So if you take a look at the chip that I showed you that Poly's group has built, over here, one thing that you don't see is that even though the chip has been miniaturized, the system that runs the chip is this thing that is behind Poly. Like essentially there's a room full of pneumatic equipment that runs a chip that is the size of a credit card, right? And if you think back to the history of technology development, this predates most of us, all of us. There were computers that were the size of a room, right? And through miniaturization, now I have a computer in my cell phone that is far more powerful than a computer that would sit in that room. Now, our thesis in building these pneumatic circuits was that wouldn't it be nice that we take advantage of the same idea of building switches that run on air so that we can provide thinking and computation to those fluidic chips. So now we would not need that whole system that is the size of a room to run a chip that is the size of a credit card. Problem is that if you want to use these inside, so if you're Poly, that's totally great, right? You've built the platform, you use the platform to run your experiments. But if you want to take the platform to Antarctica, you can't. But if you miniaturize and embed the controllers with the same chip, now you have a shot at trying to deploy your technology in point of care settings.
Thank you, Dr. Arar, for all your insight and everything. We have one more question specifically for you. So since you have experience working both at UCI and at Cal State Long Beach, what would you tell about the differences for undergraduate biomedical engineering students at each of them?
Great. So I think more than any, let me think carefully about how I want to phrase so it doesn't become rambly. I think the most important thing to be as an undergraduate scholar is proactive. I think there are different opportunities present in each school that are pretty great. So when I was a undergrad at Irvine, and then a grad student and a postdoc, I was one of 100 people in a classroom, and I think as Dr. Pilloff mentioned, I would not directly, for example, interact with the professors or even professors that I would work in their labs, right? There was a grad student who was my mentor. So that was the set of interactions. There were perhaps more research opportunities per person, but my immediate interaction was mostly with folks who were almost my age, right? Whereas in CSULB, I interact directly with the folks that are in my lab because we don't have graduate students, right? So at least in my department, we are primarily undergraduate serving department, right? So inside a classroom, the same way, when I give feedback to the students, I'm the person who's giving the feedback. It's not like a TA that is giving the feedback. It's both, it's neither one is necessarily good or bad. That's just the difference. The most important thing, I think, is that independent of where you are, you should, since I didn't go to high school in the United States, I can't make any statement about high school life in the US. But where I grew up as a high school student, there was a lot of, and I went to a very competitive school. There was a lot of support, that there were a lot of people who would chase after me and be like, hey, where is your PSET, right? In college, the expectation slowly is that you're becoming a independent learner. So sometimes we chase after student and ask them like, hey, where is this PSET? You're about to fail. But I think as you go throughout your education, you should become more and more independent, and you should proactively seek opportunities independent of where you are.
Right. And then we have one more question for you. What encouraged you to keep going to become a biomedical engineer?
I honestly don't know, to be very, very frank. I just liked, and I think this resonates with something that Mr. Dooley said as well. I just liked the projects that I worked on. Like I did not set out to be, I also, to be upfront with you, I think of myself as a bioengineer these days, even though I was first an electrical engineer and a biomedical engineer. And, I worked more on actually medically relevant technologies during my PhD. But I think what has kept me going is just the projects. And also, to be frank, a lot of good mentorships that I received in BME over EE. Like, I think a lot of my experiences in EE as an undergrad was that I was trying to get a research position, and folks would tell me to, come back tomorrow. This tomorrow would never arrive. Whereas in BME, when I showed up and said, hey, I want to do this thing, the reaction was very positive. So I wish I had a very thoughtful answer to you, but I think the truth of it is that mostly the projects that I worked on were very interesting to me, and that's why I just kept at them.
Okay, thank you so much, Dr. Arar. We
No
have
problem.
one more question for all the panelists. So what's your final advice for all of the students on the call today? If Dr. Pilloff, if you'd like to start, and then we can move in order of the panelists.
Hi. So I think my advice would be to be open-minded about learning new things. I mean, what you're interested in, and think about what you're interested in learning more about, and then just sort of search around and see where the opportunities are, and whether it's an industry or don't be sort of I would say try not to have tunnel vision, that you have to have a certain route. I'd say more really just try to pursue a passion, and learn to sort of swim in that current.
All right, thank you. Mr. Dooley, do you have anything to add to that?
Yeah, I mean, for me, it's a similar theme to what I spoke about and what the other panelists have mentioned. That if you follow that which energizes you, you're on the right path. And just keep doing that. Keep trying to, better your own self and doing things that you like to do.
Thank you very much. And last, Dr. Arar, if you'd like to add anything, go ahead.
Sure. So the last real mentor, in a professional, codified way that I had, has this big sign behind his, desk that says, "Be kind, be kind, be kind." And I think about that in my new role a lot. Mainly, very recently, my grandmother was in the hospital and it was quite scary. So I think about, being kind a lot recently. And, it's funny because a lot of times, young scholars who are, maybe a couple of years older than you show up and they're very worried. It's been really hard with COVID, and they're very worried about their grades. And it's so easy to tell them no, you missed this deadline. We can't change this, but better or worse, I've changed a lot of deadlines. I've extended all of them. Maybe that's a bad response, but I think about what would be my reaction if, for example, my grandmother was in a hospital, my entire week blew up. So if I were in a class that I was teaching, I would have missed all of my deadlines too. So I recommend for myself, I'm not giving you an advice because I'm not in a position to give you an advice. I recommend that it's to be kind, and also being kind to yourself. I think it's very easy for a lot of folks who are very high achievers to get really set in trying to have success after success. And that's a very difficult thing to maintain over a long period of time. So it's really good to try to be kind to others and to yourself, because that, I think in general, and I'm genuinely not saying this to you, I'm reflecting in what has happened to me in the past 2 weeks, essentially. I think it just makes life simpler. And that's good. That's a good thing to wish for, to kind of wish for that. So that's what's been on my mind a lot in the past couple of weeks.
Thank you so much for that. And thank you again to all of our panelists. We really appreciate you giving up your time for us today to teach us about careers in the biomedical field. For everyone watching, thank you for coming, and I hope the panel was informative and helpful for all of you. Hamid, I'll give it off to you to say last goodbye to everyone.
Yeah, all right. Well, first off, thank you, Sydney, for moderating this. Round of applause. And then, of course, thank you to all the panelists. You each provided a different sort of lens that was really helpful for all of us, as I can speak for myself in particular. So we appreciate it a lot, and we hope to stay in touch. So thank you, everyone, for coming out, and good night.